Good morning, everybody. I think we'll go ahead and get started. It's my pleasure today to introduce my friend and colleague, Mike Lewis, who is visiting us here from Baylor College of Medicine. Mike got his PhD at UC Santa Cruz, where he studied genetics and circadian rhythms in fungus. He then switched directions a little bit and got postdoctoral training with actually two leaders in the mammary gland development field, two pioneers of this field. Charles Daniel at UC Santa Cruz and then Peggy Neville at the University of Colorado. He then stayed on as faculty in Colorado for a couple of years and was recruited to Baylor as an assistant professor and is in the breast center there and is now risen to full professor in the Department of Cell and Molecular Biology. Mike has worked in kind of a range of different subjects within the breast cancer field, including signaling, Hedgehog signaling, more recently STAT3 signaling, and as well as cancer stem cells or tumor initiating cells or whatever we call them these days. I'm not sure. George. And more recently, he's really been sort of working on the forefront of the development of patient-derived xenograft models, which he's going to talk about today. These have really become a critical aspect of translational breast cancer research as well as other solid tumors. And I know there's a lot of interest here amongst our faculty in sort of getting these types of things up and running. So I thought it'd be great if he would come and talk about the development and utilization of these models that he's worked with in his lab. I've actually known Mike for most of my career. I was a graduate student when he started there in Colorado, and then we both went to Baylor around the same time to start our respective appointments. And over the years, he's really been just a very encouraging and supportive mentor and colleague for me and a number of my other trainees who trained at the same time I did. So I'm very happy to have him here and look forward to your talk. And I want to say thank you for sort of rolling the dice with March in Minnesota. You didn't get the lottery, but at least it's not blizzarding, so that's true.
Well, thank you very much. Thank you for having me. Thank you for showing up at about 8 o'clock in the morning. I have actually a theory about when PhDs and MDs get separated, and it's in college where MDs think 8 o'clock classes are going to be just fine, and they go to bed right about 9 o'clock or so. And then PhDs think classes should start around 10, maybe noon, and they stay up till midnight. So I think that's where the split happens. So I want to talk a little bit about xenografts today. I'm going to talk a little bit about Hedgehog signaling, a little bit about STAT3 signaling, but I have to disclose that I'm a scientific founder and limited partner in STEMmed and manager of STEMmed Holdings. So the work I'm going to talk about really got started with two papers that we published almost 10 years ago now. So one was showing in clinical trials that tumorigenic cells were relatively resistant to chemotherapy, so such that they were enriched after treatment. And that these cells that were left over had mesenchymal features and also tumor-initiating enrichment. And the data that we demonstrated using the stem cell markers that were available at the time, so CD44 positive, CD24 low or negative cells. If you look prior to treatment or just shortly after treatment or at the end of treatment, you can see an enrichment in the percentage of cells or the frequency of cells that have these stem cell markers. And this was corroborated very nicely with an increase in mammosphere formation. So this is a surrogate for stem cell activity. The ability to make colonies, floating colonies, under non-adherent conditions. And so we never actually could do the real study, which was to do limiting dilution transplantation to demonstrate that these were actually stem cells. But I think this is holding up pretty well as a decent surrogate for the ability to make new tumors. So the markers that were available at the time, again, the CD44-24 phenotype, and then a little bit later, ALDH1 activity, so the aldofluor assay came online, and we did a little bit of work with that. But what we found was that these markers were not always expressed. So some tumors had 44 positive, 24 negative populations. Some tumors had ALDH1 positive populations. Some had both, and some had neither. So these markers were nice, but they weren't completely selective for cancer stem cells. So we started to work on these signaling reporters that I'm going to talk about in just a second. And so we were working with cell line models at the time, and they really weren't recapitulating what we were seeing in the tumor populations. And so we asked if there's one thing I know how to do, is that's transplant things from one place to another. So I said, well, why don't we just start trying to grow patient tissue and see if we could study that. So the question we're going to ask is whether xenograft-bearing mice can tell us anything about tumor-initiating cells that we couldn't do otherwise, and whether or not they can tell us something about chemo resistance. And so we started growing these things, and to my surprise and pleasure, it actually worked once we figured out how to do it. Right now, to date, we have 62 lines, 62 independent lines representing 56 individual patients. So for some patients, we have more than one line. And these are by and large triple negative, so lacking ER, PR, or HER2 expression. But we do have a couple of ER positive lines, about half dozen or so, and then a couple of HER2 positive lines, and then of course we have the new lines that haven't been characterized yet. But you can see, so the big box here is the PDX, the little box here is the tumor of origin, and actually to date we haven't seen any real differences between what's expressed in the PDX versus what's expressed in the tumor of origin, with the exception sometimes of ER, which tends to get lost in some of the PDXs, or at least reduced in some of the PDXs. So we thought we had a pretty good model here, and actually you can see even at the level of Ki67 positivity, so the rate of cell division in the PDX in a mouse is basically identical to the proliferation rate in a patient. And so more recently, there have been a lot of people around the world that have been starting to do this successfully. And so we formed a new international consortium for patient-derived xenografts in breast cancer. And you can see these are the U.S. labs over here. These are the overseas labs. So it's largely North America, Europe, and Australia that are involved right now. But we're hoping to get people from Asia to participate too. And so all total, excuse me, we have over 500 lines representing over 500 patients, and you can see the breakdown in the percentages of ER positive, HER2 positive, and triple negative breast cancers that are represented by PDX models now. And all of these people that participated in the consortium are willing to send out their models to anybody that wants them for any reason whatsoever, no strings attached. So if you just get a hold of whoever you would like to work with in here, whoever's closest, whatever, you can get the lines from them. The one thing I wanted to point out about this is even though ER positive breast cancers represent about 70 to 80% of all breast cancers, they really are underrepresented relative to the patient population. Triple negative breast cancers are overrepresented, and HER2 breast cancers, which represent only about 10 to 15% of all breast cancers, are really underrepresented in the collections. So there really aren't that many models available worldwide for doing work on HER2 positive breast cancer. And so just because it take a long time to go through these data, at the level of genomics, Matthew Ellis has shown that genomic rearrangements and variant allele frequencies are maintained between the PDX and the tumor of origin. Transcriptomics, there's very good concordance between gene expression levels in the PDX versus the tumor of origin. The correlation coefficients are about 0.95 in general, and they'll cluster together if you do the clustering correctly. Transcriptomics, there's good stability across transplantation generations. So if you transplant from one animal to another over the course of several years, you can show that the transcript levels are the same or relatively similar across all the transplant generations. Same thing with the proteomics. We haven't shown that the proteomics between the PDX and the patient are similar yet, but that's coming down with the CPTAC projects. But there's very good stability of expression and post-translational modification, again, from generation 1 to generation 15 across transplant generations. And then Alana Whelm has shown very nicely that metastasis patterns in the PDX models are very similar to what was observed in the corresponding patient. PDX models do have some limitations, and this is just a partial list. The big one that a lot of people will bring up is that they lack a real immune system. So they lack B cells, T cells, NK cells, but they do have macrophages, dendritic cells, and other immune cells. And even though we have over 500 now, we really haven't achieved representation of heterogeneity across all of the tumor types. So particularly luminal A's are missing in those ER-positive breast cancers. Those are predominantly luminal B's, or basal, ER-positive basals as well. So, we started out this whole thing talking about chemotherapy, and we were looking at docetaxel versus anthracycline-based therapies. And I asked the medical oncologist that I was working with, so, well, how do you make the choice? She said, and I said, really? There's got to be something better than that. But anyway, so in the neoadjuvant setting or before surgery or the adjuvant setting, the first-line therapies, chemotherapies for breast cancer are taxanes like docetaxel or paclitaxel, adriamycin cytoxan or AC, and then more recently platinum-based agents have come online. So cisplatin and carboplatin have shown pretty good efficacy in different classes of breast cancer. And the decision really is now a three-sided coin. It's based largely on preference of the clinician, preference of the institution for which chemotherapy they tend to give, and there are no predictive markers for deciding which patient should go on docetaxel versus carboplatin versus AC or versus anything else, really, at least in triple-negative breast cancer. And so we wanted to test whether all chemotherapies were really equal. If you look at the clinical data, the response rates to each of those three chemotherapies are relatively similar, but they have three different mechanisms. So, given that they work via presumably three different mechanisms, we predicted that the PDX models may respond to one or the other, or not at all to anything, and that each PDX would likely have its own unique response characteristic to each of these chemotherapy regimens. And so we had a little money that we couldn't use for anything else, so we decided to put it towards a proof of concept animal clinical trial. And the basic idea behind this is that unlike human clinical trials where you can only test one drug in one patient and you have to test multiple cohorts in order to be able to compare anything, with PDX models you can put the same patient on all of the arms in the trial. And you can have as many arms as you want up to the time that the people that work for you want to take a ball peen hammer to your head. So we tried a 4-arm trial just to see whether we could do it. And Sue Hilsenbeck modeled this statistically for us based on some other data that we had. And she concluded that we needed 9 animals in the vehicle control group just to establish what the normal range was, such that you could use 3 animals per treatment arm, such that anything that fell out of that normal range would be considered a significant response potentially. And again, we used vehicle, a low human equivalent dose of docetaxel that you might use in combination with something else. Doxorubicin, this turns out to be a subhuman equivalent dose because the mice don't tolerate it very well. And then carboplatin, as near as we can tell, this is a human equivalent dose. And we decided to use 20 PDX per arm and then evaluate them for various things downstream of that. And so doxorubicin, even though it's a suboptimal dose, we do get some responses. So most of the time, PDXs really didn't respond very much, but sometimes we did get decent responses. Nothing really to write home about. So these are, the black dots are the vehicle control, the red dots are the doxor treated samples. This is cytostasis, so if it's flatlined, it will be along this line. If it continues to grow, it will be above. This is the 30% shrinkage line for RECIST criteria, and then anything below that would be considered a responder. So doxorubicin, we really haven't followed up very much after that, just because these responses weren't great. But we did get a few good responses for docetaxel. Here, this one here, and a couple that showed cytostasis. But the real poster child for this would be carboplatin, where we got 7 responders out of the collection, and a pretty good spread of things that responded really pretty well versus things that didn't respond at all. And now this is a single dose of chemotherapy given at time 0, and then we harvested and evaluated tumor growth 2 weeks later. So this isn't really what happens clinically, but it's what we had money to do at the time. And so we've done, again, we have a lot of omics on these, so we've been we're trying to establish the method really for how to evaluate animal preclinical trials. And one thing I want to point out here is that these two models here are BRCA1 carriers. One of them responded really pretty well. One of them didn't respond very well at all. And we'd like to figure out, with more models obviously, what the difference between BRCA1 carriers that respond versus BRCA1 carriers that don't respond. And so in terms of omics, we're shooting to have genomics, transcriptomics, proteomics, metabolomics, and RNA integrity score for everything so that we can have a comprehensive set of omics. But we have exome done on 33 and we have 23 pending. Turns out that this is really difficult at the analytical part because you're mixing two species and looking at reference genomes that don't necessarily have anything to do with what you're looking at. Transcriptomics is a little bit easier. We have 37 of them done. We have 19 pending. RPPA, we have so proteomics, reverse phase protein arrays done with either Gordon Mills or Dean Edwards at Baylor. We have 20 of them, the triple negatives that you just saw in the animal clinical trial. We've done those. We have total proteome on 12. We're hoping to ramp that up. Metabolomics, we have 200 analytes measured on 20, the same 20 that you just saw. And then the RNA integrity score for that set of 20 as well. And so to try to get an idea of whether or not we could come up with a differential predictor for treatment response, we looked at the RNA-seq for these guys. And these are plotted as a composite index of the fold change relative to control, and the red or pink is the p-value. So anything that's really dark red was highly significant. Anything that's gray wasn't significant at all. And then looking at the transcriptome at the RNA-seq level to see what genes were correlated, whose expression correlated with response or resistance. Same thing for carboplatin over here. And what you can't really appreciate very well is that the gene list here and the gene list here don't overlap at all. So we're pretty excited about that. And you can see that here are the carboplatin responders, and they're kind of all spread out over here in the docetaxel group. So we think we're going to have a pretty decent way of developing a differential predictive signature. And so we wanted to look at the RPPA as well. And this didn't turn out quite as nicely. So again, there's very little overlap between these proteins and these proteins, and there's really no very good signature for being able to predict response, at least at the level of RPPA, which is only about 200 antibodies. What was most interesting to us, and this has a little bit of a caveat that I'll tell you about, is that at the RPPA level, if you look post-treatment relative to the expression of whatever analyte it is prior to treatment, the response to carboplatin and the response to docetaxel were largely similar. So these are Pearson and Spearman correlation coefficients over here, the red being significant, black being not significant. Black being significant and red being most significant. Right. Anyway, these are the analytes that were upregulated in the sensitive, the resistant population. These are the ones that were upregulated in the resistant population, or the sensitive population. Get this backwards. Anyway, there's quite a lot of overlap between these two plots, and so we looked at that, and basically those analytes that were upregulated in the carboplatin responsive group, there was a 34 out of the 40 some odd analytes were shared in common, with only a few that were unique to that response. Same thing on the negative side, so things that were downregulated as a function of response were largely overlapping. And we're doing the bioinformatics to figure out what those things are. But there are a lot of things that you might expect. So survival pathways in the resistant population are upregulated. DNA repair in the resistant population is upregulated. A lot of things that we already knew a whole lot about. So as I said, the single dose, wait 2 weeks and evaluate really isn't what happens clinically. And so we've since gone on to do a multi-cycle trial where we're treating animals every week with chemotherapy. And as far as I can tell, that would be about where you would want to treat an animal based on the metabolism of the drugs and the PK/PD parameters. And so we hypothesized that docetaxel and carboplatin work by different mechanisms and have largely non-overlapping sensitivity in the PDX lines under the conditions that we tested. So we actually started a clinical trial based on this, looking at docetaxel and carboplatin together, and we're doing a co-clinical trial in the PDX models to see how well things track. And so the idea is that if you use the two of them together, that things might be better than using either one alone. And again, to test this idea, same idea, 9 in the vehicle control, 3 per treatment arm. We have recruiting the PDX patients. We're going to do this in 40 PDX models over the course of about 2 years. And then these are the 4 treatment arms. So vehicle, docetaxel, carboplatin, and then the 2 together. Same sort of outcomes that you can measure as we saw before. Basically, we get 4 different kinds of patterns. One is that it's either resistant to both, so it continues to grow either with docetaxel or carboplatin or the combination. So the combination doesn't do any better than either one alone. Sensitive to both, again, the docetaxel, carboplatin, and the combination are superimposable on one another as far as response. And if you were expecting that the combination would do better, you'd expect this line to drop more precipitously than the 2 single-agent arms. The third pattern is that PDX will be responsive to one agent but not the other, so either responsive to carboplatin and not responsive to docetaxel, or the other way around, with no added benefit of having the 2 together. And then we only have 1 out of the 15 that we've tested so far with this approach where you see some sort of an interaction. So these 2 are both resistant to docetaxel and carboplatin, but if you add the 2 together, you get basically cytostasis in this particular model. And it's the only one of the 15 that does that so far. So if that holds true clinically, you really don't get a benefit of adding both drugs, except that you have some that will respond to docetaxel, some that will respond to carboplatin, some that will respond to both, and then if you give both drugs, you get both responders. But in terms of biological interaction, there really isn't very much at all. And so this was really disappointing to the clinicians who were running this trial, but I think it's going to hold up pretty well. So if you look at the responses to multiple cycles versus a single cycle, these are ranked based on carboplatin response. The gray boxes here are the single dose results. So these are resistant, and these are the RECIST criteria. So progressive disease, stable disease, partial response, complete response. And you can see we didn't get a complete response to anything in a single dose. But in the multiple cycles, so the circles here, we did actually get some complete responses to carboplatin. But what you can see, I think, is that the response to multiple cycles isn't necessarily identical to the response in the single cycle. You might not expect that, but these were frankly resistant. That they just continue to grow. Whereas if you hit them with multiple cycles, because of the kinetics of killing, you get a little bit of initial growth, and then things tank later on. So after 2 weeks, you get it looks like it's going to be resistant, but if you actually hit it harder, you can get a pretty decent response. Same thing with docetaxel. Again, sometimes the response matched, sometimes it didn't, and in general, the response to multiple cycles was better than the response to a single cycle. The numbers that you see, we didn't have any patients that were treated with carboplatin, but we did have patients that were treated either with docetaxel or AC. And with docetaxel, it's really a pretty good agreement between the single dose and what happened in the patient. So if the patient was resistant, the PDX was resistant in the single dose. If the patient had a partial response, there was a partial response in the PDX. Where things break down a little bit is in the multiple cycle studies. So the response in the multiple cycle studies in the PDX models is actually a little bit better than what we were able to achieve in the patient. So what you get out of this depends really on how you do the study. What this says to me is that we haven't quite dialed in what really is a human equivalent dose in a mouse. So what we need to do is tweak this a little bit such that we get a dose, a route, and a frequency in mice where we recapitulate what happened in the patients. Then I think we'll have a really good set of models to study. So more exciting to us was that even though the responses were remarkably similar between docetaxel and carboplatin, we pretty much dropped the doxorubicin and AC as far as the analysis goes. You can see that they match up quite frequently between docetaxel and carboplatin, but there are some differences. So you can see this one here responded for docetaxel, but didn't really respond to carboplatin, and you can have the opposite as well. And if you look at that, we had and this includes both the multiple cycle study that I talked about and another study that has the same single agent arms. We've just kind of peeled off the single agent data from both studies for this. So we have 22 PDX models so far that have been treated with docetaxel or carboplatin so that we can get the single arm responses. And so we have 2 that responded to carboplatin only but not docetaxel. We have 3 that responded to docetaxel only but not carboplatin. 6 that were cross-resistant for both, and then 11 that were responsive to both agents. So we think that these guys and these guys will give us really pretty good discriminatory power once we get up to 40 or 50 PDXs to be able to use the omics to develop predictors for differential response. And for identifying that group of patients, which is really pretty large, where it doesn't matter what you give them. You can give them either docetaxel or carboplatin and they're going to respond just fine. But these patients, if they receive docetaxel, are going to die. And these patients, if they receive carboplatin, are going to die. And so what we'd really like to do is figure out this whole range of who should get what. And so again, the important thing here is whether, excuse me, Mona, I'm having a wardrobe malfunction. Hopefully that won't happen again. So are there any questions up to this point? Since we have a break? Yeah.
Do these models, like ER positive, do they respond to targeted therapy?
Yes, so we've done a little bit of that. So I've kind of stayed away from the ER positives and HER2 positives because at Baylor I can't throw a dead cat without hitting somebody that studies ER or HER2. So I've been relying on other people to do those studies. But for the models that we've tested, if the patient was responsive to lapatinib or trastuzumab, the PDX matches as well.
Quick question too, actually, about your gene signature that showed essentially gene patterns that were, or expression patterns that were correlated with the carboplatin response. I noticed that actually, at least I think I noticed, that the BRCA1 PDX patients models actually both had that same expression pattern or a similar expression pattern. So even the one that showed essentially cetastasis or did not show a tremendous response versus the one that had a remarkable response. Still has that same gene signature.
So the caveat that I was supposed to tell you about, I didn't tell you about. Okay. So it turns out that in RPPA, even though these antibodies are supposed to be human-specific, they're probably not. Okay. They probably just weren't tested against mouse. And so we have a lot to do to validate whether or not the RPPA signature is a tumor signature or a stroma signature. If it's a stroma signature, and this is just a response to dying cells, then that would explain what we see. What we're hoping is that it's kind of a mix between what happens in the tumor and what happens in the stroma. My guess is that because these cells are dying in similar ways, it's all apoptotic cell death, that we're actually getting similar sorts of, even though the mechanism by which the cells are dying or were induced to undergo apoptosis is different, the response of the tumor may be very, very similar.
Question, I'm sure you keep stocks of each cell lines, et cetera, the mother cell, but is there pheno or genotypic drift over time as you've looked at these cells?
I thought there would be, but we've looked at the transcriptome and RPPA for some of the models out to 15 generations, and we actually have most of our models out to 20 generations now, and we're getting ready to redo that. But at least out to 15 generations, there's virtually no drift. I thought as soon as you put these into mice, they would start to go all over the place, and it turns out they don't. So the fun thing is that I just found out last week that one of our collaborators made cell lines from 9 of these, and he's been passaging them. I think he's on passage 120 something. And so what we can do, because one of the big things with cell lines is that they don't represent the heterogeneity of the tumor of origin, and they drift genetically pretty much as soon as you put them on plastic. So we have an opportunity to actually test that relative to the patient of origin. So if we have triplets, patient, PDX, and cell line, we'll be able to see how fast do cell lines drift, and at what point do you really lose heterogeneity.
And what changes?
And what changes, exactly.
So the most meaningful clinical response is pathologic complete response after multiple cycles. So have you analyzed the bioinformatics in that subgroup that had what you call the CR after multiple cycles?
We're I'm desperately trying to get that done. Yeah. So as far as, we have ones that are just frankly resistant to whatever you throw at them. And then a pretty good spread here between things that respond a little bit, things that respond more, and then things that just go away. And for carboplatin, I didn't put these data in, but by and large, if a tumor goes away with carboplatin, it doesn't come back.
Yeah, and that's a fluke that we want to know.
Yeah, and the converse is true for docetaxel. Almost everything that has a path CR or a complete response will come back eventually for docetaxel.
Mike, you hear people say that the mouse stroma replaces the stroma of these. Is that true? And what does that mean for the drift? I mean, does that mean that the stroma doesn't have a big influence once the tumor is fully formed, or what do you think about that?
So the work that's been done in that area kind of follows on what was done in the normal mammary gland work, where if you transplant from one species to another, the host stroma overtakes the transplanted stroma, because the stroma doesn't really divide very well. Same thing happens in tumors. You transplant a little tiny fragment of human tissue that has both epithelium and stroma in it. As it grows, the stroma doesn't keep up, and so it just gets replaced.
But then you don't see drift, so then the microenvironment people are going to be upset.
So there's still stroma there. It's just mouse stroma.
It means that
Yeah.
the tumor can co-opt the mouse stroma, so it might co-opt, and then that process will continue to be important.
And we do have a significant fraction of tumors from patients that don't grow in mice. And so it may be a stromal factor that we're missing,
Right.
or some stromal tumor incompatibility that doesn't allow them to grow. If we can figure that out, that's a potential treatment.
Are you using the prolactin mice?
No, we haven't gotten those yet.
Okay.
Yeah. We've been going a different way.
Do your chemotherapies target stroma? I mean, do you see cell death in the stroma? And
I don't know that I don't think we've looked. That's a good question.
as
I mean,
a environment person.
Yeah.
Funny you ask.
So, I mean, it might be relevant.
It might be very relevant to the RPPA. Yeah. So let's see. So, as I said, if you look at the single dose, you get really pretty good concordance between patient and tumor. And when we do the statistics, it's about a 92% concordance between patient and tumor. If you look at the multi-dose and compare that to patient, because we do better in the mice than we do in people, that concordance actually falls off a little bit. But it's still pretty good. And again, I think we just need to dial it in a little bit so that we match what happened in the patient and what happened in the mouse, and use those conditions from now on. So we're pretty excited about this, and we've gone on to now work with different agents, targeted agents, in triple-negative breast cancer. And so, again, we've been working with stem cells, and we've been working with a couple of collaborators. So I'm not going to talk about this, but we've used sunitinib and crizotinib alone in combination, and I think we did 16 models, and that paper is just about ready to go. We've been using stem cell targeted agents, so Hedgehog inhibitors, IPI-926 and GANT61, that target two different members of the Hedgehog pathway. A STAT3 inhibitor that's being developed by StemMed called C188-9, and we'll come up with some other unpronounceable name for that later. And then I just got a grant to study these four agents. So a BCL-XL, BCL2 inhibitor, and you may have noticed on the RPPA that's the only one that was in common as a predictor of resistance or sensitivity between carboplatin and docetaxel was BCL-XL. The CTEP gene drug list is really unremarkable because it's a lot of things that we already know are important in cancer, so studying them really wasn't going to be my thing. But there were a couple of drugs that I thought were interesting. One was an HDAC inhibitor, and then the other one was an EZH2 inhibitor, and then we had, for other reasons, we're going to study a PARP1/2 inhibitor called niraparib from Tessera. And so what I'm going to talk about a little bit is the Hedgehog inhibitor work and the STAT3 work, and I won't talk about any of this other stuff. And so together with Jeff Rosen, we've been making basically signaling report, lentiviral signaling reporters, focused on stem cell self-renewal pathways that are important in stem cell function that are either from the literature or from our own work are important. And this is now a growing series of vectors. So the basic one is a Tata-box only. So this is a minimal or non-existent promoter, really, upstream of GFP. And so if there's, you put this in, you're expecting no GFP signal if things are working right. We have an EF1 alpha promoter-driven GFP construct, and this is a constitutive reporter that doesn't matter what it is. If it's in there, it's green. If it's not there, it's not. We have a Wnt reporter. That's the TCF-LEF. This is the top GFP reporter. We have a couple of other reporters now as well. So 3 binding sites for the TCF-LEF proteins, upstream of GFP again. M67 is the STAT3 binding site, and we think it's specific for STAT3. And we haven't seen any overlap between STAT5 signaling and greenness. And then we have a 7X Glee binding reporter that reports on activated Glee downstream of Hedgehog signaling. So Jeff Rosen's lab has been using the top GFP and other Wnt vectors, and Mei Zhang, when she was in the lab, showed that the GFP positive population grew tumors at a much higher frequency than the GFP negative population, and that these cells that were expressing GFP were relatively radiation resistant. We went on to show that we could localize these cells, and the idea was that the stem cells were going to be in a hypoxic environment, that they were going to be often some sort of a niche. And we've shown, I think, that that's not true. So if you look at the GFP expressing cells that are highly enriched for the stem cell population relative to blood vessels, you can see that the green cells are right up next to the blood vessels. And in fact, if you do the calculations, it's either 1 or 2 cell diameters away from the blood vessel. And if you look at HIF1 alpha standing in these same samples, the green cells are on the outside of the hypoxic area, not on the inside. That depends on the model. There are some models that don't follow this pattern, but by and large, for the models that we've looked at, this is holding up. And so my lab's been studying Hedgehog signaling for a little while, so we switched over to the 7X Glee. And we've looked in a couple of triple negative models where you can see, so this is MC1. This was a model that was used by Max Wisha to demonstrate that Hedgehog signaling had something to do with cancer stem cells. And it's actually a very small proportion of cells that are positive for the GFP in the Glee reporter. So it's a little less, it's about 2% or so of the cells are actually responsive to Hedgehog signaling. And let's see. So we've just recently been able to demonstrate that the GFP positive cells in both a mouse tumor, so this is tumor 505. This is a tumor that was a spontaneous tumor that came up in our MMTV smoothened model. It was the only one out of thousands of animals. So that was pretty disappointing. But you can see that the GFP positive cells are about fourfold enriched for tumor initiating capacity relative to the GFP negative cells in that model. And then if you look at the MC1 model, and this is really pretty exciting because it's the second model where we've seen this, and I'll talk about the other one in just a second. But if you look at the GFP positive population versus the GFP negative population, it's only the GFP positive population that's making tumors, at least up to 50,000 cells. You don't see tumors in the GFP negative population. So this is pretty exciting because then if you look at, you do analysis on the GFP positive population versus the GFP negative population, you might actually be able to see something in terms of omics. Whereas if you use the 44 positive, 24 negative, or Aldiflor, or a fair number of other markers, you don't see that separation. The negative cells still make tumors, but just at a lower frequency. So this should give us some really good discriminatory power for seeing what's actually expressed in stem cells. So we've been working with a lot of the antagonists. We did a little bit of work, in vitro work with cyclopamine. Really weren't very happy with that. We've done a little bit of work with vismodegib from Genentech. Most of our work has been done with IPI-926 from Infinity and GANT61. So IPI-926 is a derivative of cyclopamine, and cyclopamine comes from this corn lily plant. And if you happen to have sheep grazing, and a pregnant ewe eats this corn lily plant on the third day of gestation, this is what you get in terms of lambs. So they lack a midline, they have a single eye, they have a proboscis instead of a nose, they have cleft palate defects, and they don't typically do very well after that. So we've used a smoothened inhibitor, and we've also used GANT61 from Runatofgard that's a GLI inhibitor. And so looking at mammosphere formation efficiency, you can see the vehicle versus IPI-926 versus GANT61, both in the tumor 505 model and the MC1 model. The smoothened inhibitor actually blocks mammosphere formation really nicely. For whatever reason, the GANT61 doesn't. And you may have an idea about that, but I was a little bit surprised and disappointed at this, even though the mammospheres are quite a bit smaller in both cases. We wanted to be able to be sure that we were able to hit target in vivo, so we looked at the reporter activity in vivo downstream of both IPI-926 and GANT61, and you can see that the reporter activity in both cases is actually hit. So I feel a little bit better relative to the mammosphere formation. I don't understand it, but anyway, the reporter itself, we can shut it down either with the smoothened or the GANT61 inhibitor.
So, Mike, don't you think size versus number maybe suggest different populations of progenitor
Could
stem
very well.
George cells?
Yeah. Yes, and I may be able to talk about that. We have some work with Haida Ford that might explain what's going on there. So we've used the same kind of treatment strategy that was used in that single dose study. So looking at treatment either with a single dose of chemotherapy plus or minus a stem cell inhibitor. And again, we can break these down into 4 groups and then look at all of the stem cell assays that we might want to look at downstream of treatment. So in the studies I'm going to show you, we did a single dose of docetaxel and then daily Hedgehog inhibitor, both in the IPI-926 and the GANT61. And we're really disappointed. So looking at either the single agent inhibitor, this is the IPI-926, versus chemotherapy, there's really no interaction between the two, and the single agent doesn't really do anything at all to these tumors. So this is 3 tumors. Tumor 505, again, that's from our MMTV smoothened model. that we really expected to respond. MC1, which we expected to respond because that's the one where Hedgehog signaling seems to be very important. And then 2147 was our negative control, which doesn't express GLI1 or GLI2 to any sort of extent, which are the two transcriptional activators downstream of activated Hedgehog signaling. So we were disappointed with this. So we went on with the Gantt61, and we're a little bit more encouraged. Tumor 505, not really much to write home about for anything. A little bit of single agent activity maybe in MC1. A little bit of maybe an interaction between docetaxel and Gantt61, but really nothing to get excited about. And again, nothing in the 2147. So being really, really stubborn, I said, well, why don't we go ahead and do the stem cell assays downstream of this? And so I finally got my research associate to agree with me. And we've done this quite a lot now. So if you look at MC1, we have some unconventional, and this holds true for to a certain extent for tumor 505, but for MC1, if you have vehicle alone, you get the nice expected decline in tumor initiation as you go down in cell number. Same thing for docetaxel, and interestingly, we didn't see an enrichment in tumor initiating cells as a function of docetaxel treatment here, which we expected. But look at what happens with the smoothened inhibitor or the combination. So you plow in a lot of cells, so 45,000 cells, where you have nearly 100% tumor take rate in untreated animals, and you get nothing. You go down to a lower cell number, where you get a lower take rate in the vehicle control, and now all of a sudden you get tumors. So even though you have more cells here, fewer tumors than here, and that declines a little bit as you go down. And then the combination is the same sort of thing. No tumors with a lot of cells, an intermediate number of tumors with an intermediate number of cells, and then no tumors with the lower number of cells. And so I had them do it again, and it came out the same way. And then we also looked at the Gantt61, and the same sort of thing happened, though not in the vehicle control. So the vehicle control group, again, a nice decline. We lost this group of animals, but the Gantt61 really didn't do anything by itself. But in combination with docetaxel, again, a lot of cells, few tumors, an intermediate number of cells, lots of tumors, and low number of cells, it goes away. So it's this biphasic effect. And we think we have a reason for that we think we're targeting the stem cells directly, and we're targeting a stromal population that is impinging on these. And the idea here is that you have an inhibitory cell type that's rare, very rare, but that's capable of inhibiting the function of the tumor-initiating cell population. So that if you have an inhibitory cell here, it's capable of shutting down the tumor-initiating cells. But as you dilute out the inhibitory cell before you dilute out the tumor-initiating cell population, and that frees up those tumor-initiating cells to function. And so we have two experiments that are ongoing to demonstrate whether or not that's true. So just want to touch very briefly on the STAT3 model. Again, we've actually been showing that the reporter positivity correlates with stem cell function in cloud and low, particularly in cloud and low models of breast cancer. This work was done by a graduate student in my lab, Wei Wei. So she was able to show, again, just like with the Hedgehog, that the green population, so the GFP positive population here, grows mammospheres at a much higher frequency than the GFP negative population in both 231 cells and some 159 cells. In some 159 cells, it was only the green cells, again, that made mammospheres. The GFP negative cells never made mammospheres. So we got pretty excited about this, and so she wanted to see whether a STAT3 inhibitor would work. Unfortunately, she used static instead of the one I asked her to. Anyway, DMSO allows mammospheres to form, but if you put in static, you can block mammosphere formation in both of these models. And then in terms of limiting dilution, in both models, the GFP positive population makes more tumors than the GFP negative population, and it's enriched over the mock sorted population. I kind of like the way she did this here. But even down to a single cell in MDA 231 cells, you can get a tumor. We were really excited here. Again, the GFP positive population in SUM159 is the only population that will make a new tumor. Even up to 20,000 cells plowed in, the GFP negative population never makes tumor. So now we have two models where we have an exquisite separation of reporter positivity in the tumor initiating versus the non-initiating population. And so we think we can get either single cell resolution, or at least we can do populations and get a good handle on really what's in stem cell or not. Again, we looked in patient-derived xenografts, and this reporter works pretty well. And then here's the study that we were about halfway through. So we're looking at 25 PDX models, looking at the STAT3 inhibitor alone, docetaxel alone, carboplatin alone, or the two combinations. And again, we're about 15 models into this. And we don't really see anything as far as an effect on tumor growth with the STAT3 inhibitor alone. But in a fraction of the models, and we now have 2 models out of the 15 that do this, we're looking at recurrence after complete regression here. So you treat the tumors until they go away, and then you monitor them for when they come back. And so in this particular model, almost all of the docetaxel-treated animals, the tumors went away, and as well as the C188-9 plus docetaxel. But unlike the C188-9 plus docetaxel animals where there was no recurrence, all 11 animals that had regression in the docetaxel-only arm, all of them came back. None of them came back in combination with STAT3, and we now have a second model that does this. So we think we've got, at least in a subset of triple-negative tumors, STAT3 is very important for stem cells. In another subset, a larger subset, STAT3 doesn't seem to be correlated with stem cell activity. So I think in terms of stem cell targeted therapies, they appear to be effective in some cases, not in others, and you might expect that given the heterogeneity of triple-negative breast cancer. They're likely to fail clinically as single agents, and I think that's coming true with the Hedgehog inhibitors. They're not doing very well in a lot of solid tumors, including breast. Tumor shrinkage, though, is not an appropriate endpoint if you're going to evaluate these. If you're really only looking at 2% or 1% or 0.1% of the cells, and you kill those cells, you're not going to see tumor shrinkage very, very quickly, particularly if those other cells don't care whether there's Hedgehog signaling around or STAT3 signaling around. They're dividing for other reasons. So you may not see very much of an effect as a single agent. You may have to have a cytotoxic agent in there in order to see what you want to see. And then we think these signaling reporters are going to serve as useful markers for tumor-initiating cells, and particularly in those models where you have that nice sharp separation between tumor-initiating green cells and non-tumor-initiating green cells, non-tumor-initiating non-green cells. But we think different tumors are going to have different tumor-initiating cell populations. So you have to know quite a lot about who you're going to treat and who you're not going to treat based on their molecular features of their tumors, and potentially even the reporter expression and the behavior of the reporter-positive cells before you decide what treatment to give them. So I think this is going to be very challenging, but I think ultimately we'll find a battery of these agents that can be used together with chemotherapy. So thank you. These are the people that were involved in a lot of this work, and I need to update the slide a little bit, but these are the people in my lab. So Xiaomei Zhang made most of the PDX models that we have. Lacey, Emily, Ali are Team Xenograph. They're responsible for all the treatments. And I like to keep them very, very happy, so I take them out quite frequently so that they don't bring a ball-peen hammer into work. Wei did a lot of the STAT3 reporter work. John Landaway does pretty much everything. This has been a long-standing collaboration with Jeff Rosen, particularly with Mei Zhang when she was in the lab. Trey Westbrook for the sunitinib, crizotinib work that I didn't show. David Torty is my business partner and STAT3 inhibitor guy. A lot of people in the breast center. The PDX work is really not very easy to set up, and it takes everybody that is involved in the process to buy into it. So we've managed to get a good group together that's bought into the PDX concept. Other people at BCM, so this is the informatics group and all the omics people, working with Max Wisha for a long period of time on the stem cell work, and then Gordon Mills's group over at MD Anderson for the RPPA, Chuck Perot and Alex Pratt for some of the bioinformatics, and then for the avatars, of course, James Cameron. Anyway, I can take a couple more questions. Thank you.
I've got a question about the STAT3 work and the STAT3 reporter. How well does the reporter correlate with STAT3 phosphorylation? And the reason for asking is that it could be reading out just STAT3 activation, or even if STAT5 doesn't turn it on, typically those things will oppose each other's activity. You could be reading out the balance between the two, and that might be important to want to look in patients where you're not going to have a reporter.
Yeah, so it's kind of one of those, I forget the name of it, but it's an inequality, right? So green cells typically are phosphostat-3. Phosphostat-3 positive cells are not necessarily green. And we think that this has to do with the half-life of activation. So phosphorylation of STAT3 is probably a very transient event, and the GFP is going to stick around for 24, 48 hours. So we were hoping that they would correlate quite nicely, but they don't actually.
So, Mike, you started with the idea that the chemotherapy-resistant cells are the stem cell-enriched population. So, isn't that why you should be doing the stem cell inhibitors? Wait until you identify those subset of tumors that won't respond to chemotherapy, and then come in there?
Yeah, so this is kind of a melding of a couple of different projects. Yeah. So the chemotherapy work, we're trying to find a differential predictor.
Right. No, I'm just saying, but
right.
Your initial observation, and Jenny's too, is that the things that are still left after neoadjuvant chemotherapy are hardly enriched stem cell population. So if you're going to come in with an inhibitor, that's, you
come in after the chemotherapy?
Well, yeah, so the patients who had a complete response don't need anything. The patients who had less than a complete response need something. And if they're enriched in stem cells, maybe that's what you want to look for their response.
Yeah, so the approach that we've used up till now is to either have the stem cell inhibitor on board before chemotherapy and alternate them, or give them concurrently. We haven't tried chemotherapy, wait for a response, and then come in with the stem cell inhibitor.
Yeah, wait for lack of response.
Or a lack of response.
Yeah.
But there again, if you have a tumor that doesn't respond at all to chemotherapy, and you come in with a stem cell inhibitor that only hits 1% of the cells, you may not see anything at that point. I think the combination probably of a chemotherapy that works and a stem cell inhibitor that works is probably going to be the key. But then the trick is knowing which chemotherapy to use and which stem cell population you have in there. So one of the things we've done is we've changed the color out on several of these vectors so that we can put them in at the same time and evaluate different populations for whether or not there's the stem cells. So you could have potentially a Hedgehog positive, STAT3 positive population that's a stem cell compartment in one, and then a Wnt positive, Hedgehog negative population in another tumor model. So hopefully we'll get the money to actually do that in all of our models, or as many as we can. One of the other limitations with PDX models is they don't like to be dissociated very much. So we have to be very careful when we do the lentiviral transductions not to dissociate them down to single cells, because then they won't retransplant. So we have to do them kind of a slurry, kind of a minimal dissociation, and then we get about 30 to 50% of the cells infected.
Very interesting models.
Thank you.
What I'm curious about is that in most of the cancer cells, the signaling molecules generally, localize differentially compared to the normal cells. And I was wondering if you have seen any changes in these models with respect to that kind of thing.
We haven't. With respect to Hedgehog signaling, there's a lot of work now done with primary cilia that the hedgehog signaling happens in the primary cilium and actually depends on the primary cilium for activated signaling. We haven't really looked in the cancer models, but presumably there would be differences. So potentially, particularly in smoothen mutants that don't localize to the primary cilium but are activated anyway, we would predict that we would see differences in localization there.
I'm wondering if they persist in these models.
Haven't looked. I would guess based on the similarities in gene expression and protein modifications and things like that they would hold up between patient and PDX, but I don't know that for sure. Well, thank you very much. I appreciate it.